presenter is Dr. Professor Shimoda. The title of his talk, Autoimmunity Against Biliary Epithelial Cells. Shimoda, please.
Thanks, Chairs, and thanks, Dr. Shitanaka, for giving me an opportunity to speak here. And I have no COI. And this is the process of PBC progression we suggested. In the beginning, Toll-like receptor ligands stimulate innate immunity, and activated NK cells kill autologous biliary epithelial cells, and they release autoantigens. And then autoreactive T cells are activated, and again, they kill biliary epithelial cells. And organ specificity in PBC is here, hydrophobic bile acids and toll-like receptor ligands. They modify PBC phenotype. And at last, innate immune cells and acquired immune cells and target cells consist of chronic non-suppurative destructive cholangitis. Then today's contents is here. The first is biliary epithelial cells in PBC. As mentioned, main target of PBC is the epithelial cells of intrahepatic small bile ducts. And they are always exposed by hydrophobic bile acids and intestinal bacterial components such as dumps and pumps, because biliary epithelial cells in small bile ducts do not secrete mucin. Then they are naked, and they are so sensitive to danger signal. First, we performed PBC mimic culture system. From explanted liver, we selected EpCAM-positive biliary epithelial cells, and from explanted spleen, we purified immune cells. And immune cells and autologous biliary epithelial cells were co-cultured ex vivo in the presence of toll-like receptors or hydrophobic bile acids or cytokines which mimic PBC environments. The question here is, do hydrophobic bile acids influence biliary epithelial cells? The answer is yes. Hydrophobic bile acids, such as glycine-conjugated chenodeoxycholic acids, induce biliary epithelial cells to produce high expression of immune molecules in the cell surface, such as HLDR molecule or CD40 molecules. And previously, we found biliary epithelial cells produce cytokines and chemokines. This is the result of cytokines and chemokines panel. Without stimulation, biliary epithelial cells produce IL-6, GRO-alpha, GCP2, and IL-8. And in the presence of toll-like receptor 3 ligands, biliary epithelial cells produce Th1-induced chemokines like MIP1-alpha, MIP1-beta, Lantis, and IP10. Then we focused IL-6, IL-8, and IP10. Again, hydrophobic bile acids induce biliary epithelial cells to produce such cytokines and chemokines from biliary epithelial cells. And we performed migration assay of lymphocytes toward autologous biliary epithelial cells. Hydrophobic bile acids treated biliary epithelial cells attract many lymphocytes when compared to control. Then hydrophobic acids induce biliary epithelial cells to produce high amounts of chemokines and inflammatory cytokines to recruit and activate lymphocytes. In summary here, biliary epithelial cells acquire PBC phenotype in the presence of hydrophobic bile acids and toll-like receptor ligands. Next, move on interferon gamma in PBC. In this PBC mimic culture system, the next question is, do immune cells produce cytokines in the contact with biliary epithelial cells? And such cytokines influence biliary epithelial cells? To answer this question, we prepared 4 PBC samples. Biliary epithelial cells immune cells were contacted with biliary epithelial cells. After the contact of biliary epithelial cells, we found 79 genes were downregulated and 124 genes are regulated by microarray. And when biliary epithelial cells were stimulated by hydrophobic bile acids and toll-like receptors, we found 101 genes were downregulated and 80 genes were upregulated in immune cells. And we compared between this panel and this panel. And we found only 5 genes are upregulated after the contact of biliary epithelial cells, that was stimulated by bile acids and toll-like receptors. And among these 5 genes, interferon gamma is here. Chronic expression of interferon gamma leads to murine autoimmune cholangitis is a female predominance. It was reported in 2016. And in this model, they have AMA positive and they have portal inflammation, like here. And previously, Dr. Harada reported that PBC bile ducts had high expression of CX3CL1 fractalkine, that is a chemokine and cell adhesion molecule. And in the presence of interferon gamma and hydrophobic bile acids, biliary epithelial cells produce high amounts of CX3CL1. And anion exchanger 2 exchange HCO3 minus and Cl minus. Then it acts like a cell protection by bicarbonate umbrella. And interferon gamma and hydrophobic bile acids decrease AET expression from biliary epithelial cells. And we found NK cells kill autologous biliary epithelial cells in the dose dependency manner. In case of a low frequency of NK cells, they don't kill biliary epithelial cells, but they produce interferon gamma, and this induces biliary epithelial cells to express high amounts of HLA class 1 expression. And after interferon gamma exposure, biliary epithelial cells become resistant against NK cells attack, even though there are many NK cells. And in the beginning, when there are many NK cells, they kill autologous biliary epithelial cells. Then biliary epithelial cells release microparticles that contain autoantigen, PDC2. Then autoreactive T cells proliferate in the presence of microparticles, and activated autoreactive T cells produce interferon gamma. Then interferon gamma regulates both innate and acquired immunity, PBC. Interferon gamma have a hierarchy, choose the hierarchy in the innate immunity and acquired immunity. In summary, interferon gamma produced from NK cells and T cells modulate both effector and target cells in PBC. Next, move on GBUS and PBC mimic culture system, even though Dr. Gaushin said GBUS is disappointed. It was reported that IL-12 pathway is implicated in PBC by GBUS. In this model, APC was stimulated by Toll-like receptor ligands. Then activated APC release produced IL-12, and with a direct cell contact, CD4 positive T cells activated. Then they produce interferon gamma, and interferon gamma stimulate APC again. And now we found interferon gamma stimulate biliary epithelial cells to produce chemokines. Then the question is, genetic factors influence interferon gamma-induced chemokine production from biliary epithelial cells. We prepared 3 PBC patients. Biliary epithelial cells are stimulated with interferon gamma. Then we found 359 genes are upregulated, and 26 genes are downregulated using the microarray system. And among these genes, STAT1 and SOX1 is selected because from the GBUS PBC catalog. Then we evaluated the STAT1 and SOX1 expression by immunostaining. Liver biopsy samples from obstruction and PBC were prepared. And as shown here, STAT1 expression in PBC bile duct is increased, and SOX1 expression is decreased in PBC. Again, do genetic factors influence interferon gamma-induced chemokine production from biliary epithelial cells? Till now, we cannot find any differences of IP10 production from biliary epithelial cells between PBC and chemokines. But in PBC mimic environment cultures like interferon gamma and hydrophobic bile acids, they promote biliary epithelial cells to produce high amounts of chemokines. Environment factors, but not genetic factors, influence interferon gamma-induced chemokine production from biliary epithelial cells. Then next, we focus to APC, monocytes. Previously, it was reported that Toll-like receptor ligand-stimulated monocytes produce high amounts of cytokines, and PBC monocytes produce high amounts of these cytokines and chemokines. But unfortunately, IL-12 were not detected. Then we must have experiments deeper here in future. And next, we focused CD4 positive T cells. Do genetic factors influence IL-12-induced interferon gamma production from T cells? The answer is no. Till now, we cannot find any differences of interferon gamma production from T cells, which is stimulated by IL-12. Maybe genetic factors don't influence IL-12-induced interferon gamma production from T cells. But we found there are many autoreactive T cells in PBC liver. These autoreactive T cells produce high amounts of interferon gamma. And we found biliary epithelial cells chemokine production in the presence of cell contact of autologous immune cells, there are many high production of chemokines in PBC patients when compared to control disease. Then in summary, both genetic and environmental factors may contribute to the PBC pathogenicity. Here, innate immunity may be a kind of genetic factors involved, and target cells and autologous autoreactive T cells acquire immunity, environment factors contributed. The last, where UDC works in PBC. Do they work on biliary epithelial cells? The answer is yes. Biliary epithelial cells produce chemokines in the presence of interferon gamma and hydrophobic bile acids, and UDCA regulates this production. Does UDCA regulate autoimmunity? The answer is no. Autoreactive T cells proliferate in the presence of autoantigen, and they produce interferon gamma. UDCA does not regulate the proliferation or interferon gamma production. Does UDCA work in innate immunity? Do they regulate monocytes or NK cells? Innate immune cells produce interferon, type 1 interferon and type 2 interferon in the presence of toll-like receptor ligands and bile acids, hydrophobic bile acids. However, UDCA does not regulate these cytokines production. Then UDCA regulates biliary epithelial cell function in the presence of hydrophobic bile acids. Then this is our conclusion. PBC is namely an interferon gamma disease, and innate immunity and acquired immunity contribute, and target cells contribute to the PBC pathogenesis. And UDCA works on target cells. And in case of UDCA refractory patients, we must pay attention in innate immunity and acquired immunity in future. Thanks so much.
Elegant, of course. You're the world's expert in isolation of all the liver subpopulations, and I always congratulate you. Do we have any questions? So, Doctor, let me ask you a question. In this presentation, I didn't hear about the role of sex or estrogens. In your opinion, can you explain what the role of estrogens or of female sex might be?
I have no idea for the sex hormones, but as you know, usually female hormones have effects for the autoimmunity. And we must treat immune cells with sex hormones. Then in future, we evaluated what happened. Yeah.
I wonder if it goes through gamma interferon, that estrogens alter the levels of gamma interferon. And in this system, you're already adding gamma interferon, and so you've skipped the in vivo aspects.
Yeah, I will try to evaluate interferon gamma, yeah.
Other questions? Please.
I have two questions regarding your ex vivo study to the for the potential interpretation of the clinical issues. Okay, so how do you think about the before and after bile duct loss? Okay, your study is focusing on the role of Beck as a kind of the necessary and in some instance sufficient target of the diseases. But after bile duct loss, what happened in the kind of the inflammatory milieu?
After the bile duct loss, chemokine does not work anymore because biliary epithelial cells don't produce chemokines to attract lymphocytes or like that. Then anti-IP10 antibody or anti-fractalatan antibody did not affect in PBC pathogenicity.
So your hypothesis is before and after as a kind of the tremendous changes should be occurred in the inflammatory milieu, right?
After the bile duct loss, but we must evaluate acquired immunity deeper, I suppose, yeah.
Okay, and the final question is, you mentioned that the UDCA unresponsiveness, but in the last week EASL, there are so many papers describing a very potential effect of the PPA alpha delta agonist or FXR agonist or something like that. You finally mentioned that in case of the UDCA unresponsive patient, the other drugs should be targeted to the innate immunity or something like that. Do you believe in so in case of the PPA agonist or something?
I suppose these agonists, they have an effect to immune cells because they activate NF-kappa B. Then maybe these fibrillate or like that target or immune cells, I suppose, yeah.
Thank you, always. Very nice work. And my question is about NK cells and biliary epithelial cells interaction. And now I understand that PBC is basically an interferon gamma disease interaction mediated by interferon gamma. You showed the data about NK cell in vitro co-culture system with Beck, and some kind of microparticles are produced from NK cells. Is that correct? And microparticles are containing some kind of core or autoantigens or autoantibodies. And what's the role of microparticles in vivo in PBMC disease settings?
First, microparticles is released from biliary epithelial cells.
Biliary epithelial cells,
Yeah.
okay.
And in vivo, maybe these microparticles activate autoreactive T cells. Yeah. Then activated autoreactive T cells kill biliary epithelial cells again, and they release interferon gamma. Yeah, I suppose.
So the microparticles can be taken up by some kind of APCs or directly influential on the autoreactive T cells. Which way?
It depends on, I have no idea now, but yeah.
Okay, thank you.
Okay, are there any comments, question? Again, thank you very much for Dr. Shimoda.
Thank you so much.
So thank you. Yeah, no. So thank you for attending this session.
Thank you very much, chairs and lecturers.